Good afternoon. So, last class we discussed about laser capture micro dissection. So far we discussed and also we thought that microscopy is to help us visualize, the structure of any samples. But after, the laser capture micro dissection, we are not only going to see the structure of the samples, but also we can able to isolate the cells, which you want. So, why do you want to, use this protocol or this technique? We already, discussed that because the previously, when you dissect the samples to separate from healthy tissues, there will be a, contamination because imprecise, dissection, manual error, and also very, contamination. When you separate the healthy tumor cells or healthy infected cells or malignancies, when you separate them, there will be a contamination. Either it is cancer cells or it's in the, healthy. So the subsequent, experiment when you are doing this, you cannot, like believe that the whatever results you are getting is purely from, either one. Definitely there will be some contamination. So that the investigation or research purpose, so sometime it will be confusion. So to overcome all this, instead of separation of the tissues from the heterogeneous mass of tissue samples, using this laser capture micro dissection, you can able to isolate a single cell from heterogeneous mass of cells, so that it is a precise and which cell you are going to isolate and what is the morphology of that cells, or a group of similar morphology of samples. When you isolate, before you isolate the cells, you can take a picture, and also after you isolate, you can take a picture. So now the photograph will give you evidence that what is your source of sample. So when you go for subsequent analysis, like isolate DNA, RNA, or protein, and then go for some downstream application, how this helps. That is the main, advantage of this laser capture micro dissection. So it is the technique is as discussed. It's very simple. It's either it's a light microscope or fluorescence microscope. On top of that, it attached some few components, that is arms and the material storage and because thermolabel, polymer caps, so the and xenon or mercury lamps. So this will a lot of other things, laser. So this will all attach to the fluorescence microscopy or bright field microscopy. It will be called it is laser capture micro dissection microscopy. So it's very simple, and but it is the process, tissue samples, regularly how we are processing samples for light microscopy and fluorescence microscopy, we can able to do that as well. And then you can stain it if based on, you can isolate the cells based on the structure, then you can use a HNE staining, or you wanted to isolate the cells which express based on the particular protein of your interest. So that means you can use a fluorescence microscopy. So now you are going to isolate the cells. So we discussed that how we are going to isolate the cells. So the next is the slides. We can use both either frozen tissue section or a paraffin section. So based on that, we can use superfrost plus if you are using frozen section, or you can use amino acids, silane or silicon coated slides. That will be the cells, the sectioned cells will be attached to the firmly on the slides. So this is like, what are all the chemicals we need, and we discussed already in the fluorescence microscopy and bright field microscopy. Of course, then the storage is very, very important. Most of the time we are not going to, do all the experiments the same day. Sometimes we can, cut the sections, and then we cannot go for further experiments. So meantime, we have to store it. So how we can store, and if it's a frozen section, so we can place them in -20, or if it is a paraffin section, you can store it in room temperature. So as we discussed, that's hematoxylin eosin staining. Already we discussed this in the light microscopy. This is exact the processing time. So this is like first you have to deparaffinize the sections, or you have to remove the OCT medium, that is optimal cutting temperature, that the medium, embedding medium. And then now your sections stick onto the slides that you can use, to stain with the hematoxylin and eosin, and then you can again wash it, and then air dry and observe under the either fluorescence or bright field microscopy. Then if you wanted to go for laser capture micro dissection, then you can take a sections according to the use of either bright field or fluorescence microscopy. So during this time, it is very, very important when you process your time. What are all the factors which affects the recovery of nucleic acid? So our main goal is to isolate the cells. Is you have to isolate DNA, RNA, or protein for subsequent analysis. So that is our goal. Hello, can you mute yourself? So, what's the quality of a sample? The main, our goal is, if you really wanted to isolate the cells, is based on the morphology, based on the structure, how it looks. So that if that is the case, you have to make sure that the process is, good, and also when you take a sections, it has to be thin sections, that is 5 to 10 micron thickness, and also you can see properly, you can stain it, and you can visualize the structure, and then you can isolate. So that the morphology is important. If morphology, based on the morphology, means the process. Also at the same time, the capture success. So that is also very, very important. When you isolate the cells is another important. Also, during this process, the maintenance of molecular integrity. So, that is what you are going to do the subsequent analysis. These are all dependent on numerous variables, such as how to fix it, and what are all the methods you use, and which how long you can fix the samples, and how quick you are going to fix the samples, and how do you process your samples, whether it is a chemical fixation or cryofixation. If it is a chemical fixation, what are all the process, and how, in the cryofixation. So these are all very, very important when you are taking care, during the process. During this process, fixation is to preserve your samples. Next is the dehydration. So that is, to facilitate, your processing for, the following steps. So this will give you two important reasons for laser capture microdissection, the dehydration. One is moisture impedes the film wetting and capture. As we mentioned earlier, that when enable the laser beam that will melt the polymer caps, that is a thermolabile caps, polymer. Once it generate heat, the laser generate heat that will melt the polymer, and the melted polymer is sticking onto the sections, then you can pull it out. So the transient, the temperature is important because that helps to melt the, tissue samples. So if the sample is wet, if you are not properly dehydrated, like once it touches on the samples, the transient heat will be, immediately cooled down. Or it evaporate the tissues, so the section will be different structure then. So it is important this that impedes the film wetting, and the capture success is very poor. Also, if we are not properly dehydrated, the RNA degradation is most rapid in a hydrated sample through RNase activity and auto degradation. So that is the main study of this. Another one is a graded ethanol solutions of, you can use a 75, 95, and 100%. Also use a fresh 100% ethanol for all the samples when you process, during the dehydration process, you can, use a proper graded ethanol so that you can completely remove the water content. As we discussed, that is important, the tissue morphology, capture success, and maintenance of the molecular integrity, right? So this application, however, introduced methodological hurdles that have not been encountered with more conventional molecular analysis of whole tissue. So this is, we including tissue processing, staining, extraction methods. So based on so far, like our goal is the laser capture microdissection is to isolate the cells from the heterogeneous mass of cells. So our goal is to isolate DNA, RNA, or protein. So now, what are you going to do with this? What is the purpose of, isolating a cell is, a question. So our goal is like, for as a physician, as a dentist, they wanted to suspect something in the, tissues or organs. They will do the biopsy and test for, go with the bright field microscopy or, fluorescence microscopy, and they use this for diagnosis purpose. Whereas as a researcher, you have to, learn more about what is going on with this, organs, what is going on with the tissue, what kind of cancer that, and whether it is early or advanced, and early stage, how this cells looks like, and when get old, what happens that. So if you wanted to study this, and also what are all the molecular and biochemical changes happen between the healthy versus malignancy or tumor cells. So this will you need to in order to understand further this cutting edge technology. Now they use many methods apart from photoscopy, Western blot analysis, ELISA, affinity column chromatography, and other estimation, enzyme interactions. There are so many techniques, we can able to analyze, different things. But, like in those days, like 10, 15, 20 years back, we have to go with the sequencing, manual sequencing, and, to identify different genes is very, very tough because, a lot of time consuming and things. After, the sequencing, DNA, RNA, protein sequencing technology evolved, then it is easy to analyze thousands of genes simultaneously. So that is the downstream application. You can go what are all the genes are upregulate or downregulate response to either cancer cells or healthy cells or tumor cells or infected cells. So this way you can able to understand further for your treatment options as well as to get more knowledge for research purpose or future treatment options. So now that is our aim of these laser capture micro dissections, goal. So here, this represent, the patients admitted in the hospital, and we do the, biopsy samples, and you process and take a sections and visualize the structure of the cells. And now you wanted to isolate this, then cells to isolate, then you can isolate either DNA, RNA, or protein. If you isolate a protein, then you can do protein sequencing, protein microarray analysis, 2D gel electrophoresis. So this will give you, what are all the proteins are expressed, things. Whereas the microarray that will gives you what are all the proteins are expressed or, whether it is increased or down regulated. Similarly, when you isolate the RNA from the cells, then you can do RNA sequencing, RNA splicing. There are so many techniques which you can able to analyze what are all the RNAs are expressed, down regulate or upregulation. Also, if you have like DNA, then you can do 16S rRNA sequencing to identify if there is any bacterial, analysis. You can go that. So there are different applications you can use that is based on what nucleic acid is isolated from the samples. So as I mentioned that, the laser capture micro dissection technique, you can isolate a single cell from heterogeneous mass of cells. So that means the number of cells is required to isolate DNA, RNA, protein. Not just one single cell, DNA or RNA is enough to analyze all this technique. So how do you achieve when you collect more than one cells? So when you observe under the light microscopy, you can pick a similar structure of cells and then collect them in the tube and analysis. Or you can recall or any other sources that you can able to isolate the cells and you can use it. So so now, if you really wanted to isolate RNA, means when you look at this, different microdissected cells is required. For example, if you wanted to study, construct a DNA library, it has to be 25,000 cells. to 5,000 cells. If you really wanted to study gene expression analysis, 1400 RNA, like PCR, RT-PCR, that is a real-time polymerase chain reaction. You can use, different cells, RT-PCR, RNA, 100 cells per reaction it require. Then Western blot, if you wanted to study, that is, 500 cells from 2,500 cells. Then if you wanted to analyze a 2D gel electrophoresis, that require more cells. So this will gives you, some examples that, if you really wanted to study further with your samples, you'd require a lot of tissues and a similar structure of tissues so that you can easily, isolate the cells. So now, already we discussed that now. So we have now isolated protein, for example. If you isolate a protein from the dissected cells, then you can do all these, methods to identify a protein if it is a new protein expressed in that cell types. So one is you can estimate the protein concentration and characterize the proteins, and then purify the proteins. This way you can understand further what is going on with that particular disease. So this is, the application. So with this technique, the Cancer Genome Anatomy Project at NIH relies on microdissection in the search for the molecular progression of cancers, with the fundamental objective to identify genetic differences between normal, pre-neoplastic, and cancer tissues by comparing and contrasting expression profiles from microdissected regions in the same patients. So our goal is, this technique will help us to understand further the fundamental objective is to identify genetic differences between normal, neoplastic, and also cancer or malignancies tissues by comparing and contrasting expression profiles from microdissected regions in the same patients. So what are all the applications? So the gene mutation in tumor often correlate with the prognosis and the therapeutic response. So particularly the cancer cells, if we really wanted to study how the mutation, the gene mutation or single nucleotide polymorphism, due to that changes, how, it developed the tumor. Also, we can able to understand further with the prognosis and also the therapeutic responses. So if you wanted to, when you grow a cancer cells in vitro, then if you do some chemotherapy, treatment, and then you can analyze which one is the best method and how this chemotherapy helps and get them, how it, affect the cancer cells, but not with the healthy cells. So this kind of, knowledge, you will get it from this. So the efficient detection of a gene mutation is becoming increasingly important in the pathological diagnosis. Also the classification and the treatment of tumors. So the next is a cancer chemo prevention. That is the so-called intermediate endpoint biomarker can serve as a reliable surrogate at the target site to monitor success of chemo prevention of cancers. So the so-called the intermediate endpoint biomarker can serve as a reliable surrogate. The selection of an, intermediate endpoint biomarker which requires that there is an consistent association between the biomarker and the cancer. And that biomarker modulation should be observed after chemotherapeutic intervention. So usually like when the drug target, so when you have a new drug, how it target the cells. So once it targeted, how the interaction of the chemotherapy chemicals and also that cancer cells, how it is interact and how it is a prevent, that you can understand further with using when you isolate these cells. Also, biomarker discovery is very, very important to predict in advance that you can expect these things. So that is biomarker. So laser capture microdissection can be applied for biomarker discovery in multiple human tissue types and organ systems. So the LCM is a combination with DNA transcriptome. Profiling has been successfully used to identify differentially expressed genes. So that is another, transcriptome profiling that can usually differentially express the genes in the cancer cells. Also, the important application for molecular techniques are not only just for cancer or tumors, but also infectious diseases. So we can use this technique, for routine histopathological diagnosis, also southern blot hybridization, like in situ, fluorescence in situ hybridization analysis, that is a FISH technique. Also PCR you can able to do. So one is, the Mycobacterium tuberculosis, that is usually, it is dermatology. Infections with Mycobacterium tuberculosis and Mycobacterium marinum are the most common type of myobacterial infections. That the second one is the Treponema pallidum. That is a spirochete, is capable of invading numerous tissues, with skin or mucosa being the first site of inoculation in most cases. Next is Borrelia burgdorferi. That is another things that help the spirochete being associated with disease such as scleroderma lymphomas. And then the Leishmania species, that is a cutaneous and visceral leishmaniasis, are endemic in many regions. Clinically, the patients carrying the parasitic infection typically present with the papal nodule or tumor. So these are all the basic, the downstream application that, which helps to identify novel genes, diagnosis purpose and also treatment options, so that, the isolation of cells is, giving you a reliable, treatment options. So with this, we completed the laser capture microdissection technique. The next one is scanning electron microscope and the electron microscope. So the next is the scanning electron microscope. So why do you have to use, scanning electron microscope or electron microscope compared to light microscope or fluorescence microscope. What is the advantage? So that's we'll see one by one. As we discussed this, the microscope is an instrument for viewing objects that are too small to seen easily by the naked eye. So that is the purpose of our, microscope. So we discussed about this, like, less than nanometer, you can able to see through electron microscope. So the timeline for electron microscope is, 1897. First, J.J. Thomson, he discovered the electrons. In 1926, Bush, he, find the make a magnetic or electric fields as lenses. In 1929, Ruska, he made his PhD thesis is related to magnetic lenses. So first in 1931, Knoll and Ruska, first commercial EM was built. Then 32, Davison and Kalbrick, invented that the electrostatic lenses. Then 34, Dreist and Mueller. Then the EM surpasses light microscopy. In 1939, Von Boris and Ruska first commercial EM was built. So after that, 0.1, 0.2 resolution, that is different, universities, they identify simultaneously this kind of informations. 1986, then A. Crew, he's from Chicago, he developed 0.3 nm resolution probe practical field emission gun. Also, Ruska et al., he got a Nobel Prize for this during 1999. So he can achieve 0.1, now 0.05 nanometer, also now able to, analyze. So our goal is, the scanning electron microscope is visualize the magnifying images of your surface of any samples. It's not like a, light microscope. So imaging, the scanning electron microscopy, transmission electron microscopy, and focused beam electron microscopy. So mainly we can see a diffraction. That is, two different diffraction. One is backscattered electron diffraction. The other one is area electron diffraction. And also the spectroscopy, X-ray energy dispersive, also electron energy loss Auger electrons. That's another, you can able to attach this signal to the electron scanning electron microscopy. You will analyze all these details. So what is the scanning electron microscope? The scanning electron microscope, it's very, very important. Because this electron microscope, you cannot be able to see, some images because it is very, very small, so that it require electron microscopy so that you can easily visualize, evidence. Even though there are a lot of processing and chemical and time consuming, but once you get this, it is very valuable. Informations you will get it from this. So then, how do you take a picture, how you can process, and these are all another interesting, topic. We will discuss that. Also, electron beam samples interactions, how the electron and the sample is interact with each other and gives you, um, images. So, signals that can be used to characterize the microstructure. So that is, depends on when the electrons go and hit the samples and bounce back with, backscattered electrons, secondary electrons, and also X-rays. If you have an appropriate, receiver, then you will get all these, things. Also, the component of the SEM is also what are all the components that are important for scanning electron microscope. So here is also the resolution is very, very important. So the advantage of the scanning electron microscope, when you look at this, the range of resolution, for light microscopy, 100 to 200 nanometer. Whereas in the SEM, it is, 10 times lower, or 1.5, 1,000 times, 1.5 to 5. So the magnification when compared to light microscopy, the fluorescence or, other than the bright field microscopy, it can go up to 300,000 times or above. So the improvement in resolution and depth of the field, that is the most important thing when you look at. Also, the atomic force microscope or scanning force microscope, that's another, physical properties of the microscope. There is another one, scanning tunneling microscope as well. So mainly, what you can get from the scanning electron microscope. So the topography, morphology, composition, crystallographic informations. So topography is the surface feature of any object. So here the scanning electron microscope, not only living tissues when you are processing, but also you can, observe any material. So our goal is the scanning electron microscope is to visualize the surface of any objects. Also, you can able to see the size and shape of the sample. Not only the structure you can able to analyze, but also, you can measure the elements and compounds, how on the surface it was it deposited. So that also you can analyze using this scanning electron microscope. Also, the X-ray crystallographic technique that will help you, how the atoms are arranged in the object. That's melting point, reactivity, and hardening. So the scanning electron microscope, the image is seen in 3 dimensions. It also has a high magnification and at the same time high resolution, so that you will get a clear image. The specimen is coated in gold and the electron bounces off to give you the exterior view of the specimen. So our goal is the electrons will go and hit the samples and bounce back. That will give you the topography of your samples. Always the electron microscope is in black and white. Do you know why? Because it produces electron as a light source. So the scanning electron microscope, which provides the user with a magnified image of the specimen showing details not visible with a light microscope. So that's why we are going to use scanning electron microscope. The one which you cannot able to see through the light microscope, then you can use to visualize in electron microscope. Specifically for surface structure, you can use scanning electron microscope. So the scanning electron microscope generating an electron beam, scanning forth and back over the sample. So that is, electrons are bounce back, as I mentioned earlier, so that you will get a clear images. Due to this interaction between the beam and the sample, several different signals are produced, which providing the user with detailed information about the surface structure. So here, when the samples, put them in the chamber, when you generate the electrons, so it will pass from the light's electron source to go and hit the samples. And instead of penetrating the samples, it will bounce back so that you can able to see the surface of the samples. So components, of a scanning electron microscope, it is not like a light microscope or fluorescence microscope. This is little complex and expensive. So this parts is similarly scanning electron microscope is with fluorescence microscope. So one is like, the column. So I will show you next with the picture. The column is built for, to protection for the electrons. It does not, come out exposed to humans. And also the electron path should be narrow and hit the samples. So that is most important thing. So the electron source to produce electron and the magnetic lenses to demagnify the beam, also the apertures to define the beam, prevent the electron spray. So vacuum systems is very important for scanning electron microscope with the cold pumps and produce the vacuum. So the signal detection and display, which consists of detectors which collect the signal. Also electronic, which produce an image from the signal. So the electron beam and the specimen interaction. So the scanning electron microscope, which produces electron, is a light source. Whereas light microscopy, the visible light is a light source. For fluorescence microscopy, the fluorescence lamp, xenon and mercury gives you the light source. Whereas here, it gives you the light source is electron. So the electron is, once it go and hit the samples and bounce back, that time it will produce the interaction between the electron and the samples on the surface that will reflect and bounce back with the different signals. If you have a different signals detector, you can able to see all these signals. One is, backscattered electrons, secondary electrons, X-rays, so these are all very, very important. Cathodoluminescence. So all you can able to detect if you have these instrument along with the scanning electron microscope. So the electron is assumed that it will go from the electron light source and hit the sample and then bounce back. If it's bounced back with the electrical information you want, you can use this data. If you observe the secondary electrons, so you can use to analyze the topography, that is a surface. Also, auger electron, if that signal which gives you the compositional informations. Also the X-ray that gives you through thickness, and also you can analyze composition informations. Then the backscattered electrons, if you really wanted to measure atomic number, and also it gives you the topography informations. So based on this, this figure in this illustration shows that not only you are seeing the structure of any sample, but also you can able to get more information on different elements based on how what kind of signals you are getting and identify. So the electrons, as know that it is the light source, and it will we know that it is go hit the samples and bounce back, and then you can see the images. So the electrons are generated in the electron gun, that is a tungsten filament, that is serves as the source of electron in scanning electron microscope, which generate electrons and will be focused in front of the anode. To move the electrons down the column, voltage differences between the tungsten filament and the anode will be applied. So then the voltage differences, we can calculate the accelerating voltage, and can be varied between 0.2 to 40 kV, which will gives you more energy. So the beam of electrons will be condensed and focused as a fine spot on the specimens by 2 to 3 electromagnetic lenses, which is located in the microscope column. Also, the two main functions of the first two lenses, that is, one is a condenser lens and control the beam current. Also, the final size of the area illuminated on the specimen, that is a spot size, is also by these condenser lenses. So the third condenser lens also will be used primarily to focus the beam of electrons on the surface of the specimens. The interaction of the electron beam with the sample or specimen, different signals, some backscattered, secondary electrons, X-rays, which can be detected. So the interaction between the electrons and the samples are used to obtain the information of the sample, either topographical, compositional, or elemental or structural samples images, which are all depends on what kind of signals you are used. Secondary electrons. The secondary electrons are generated from the collision between the incoming electrons and the loosely bonded outer electrons. So this is always called as low energy electrons. So the secondary electrons generated close to the surface escapes, that is, you will get in the topographic informations. So the number of secondary electrons is greater than the number of incoming electrons. So then backscattered electrons, if you get these electrons, a fraction of the incident electrons is retarded by the electromagnetic field of the nucleus. And if the scattering angle is greater than 180 degrees centigrade, the electron can escape from the surface. Another signal is X-ray, that is photons, not electrons. Each element has a fingerprint X-ray signal. Also, the X-ray is poor special resolution than backscattered electrons and the secondary electrons, which relatively, few signals which are emitted and the detector is inefficient. So the X-rays is the relatively long signal, collecting times are needed. So short with the secondary electrons, mainly topography, low energy electrons, high resolution surface signal depends on the curvature. So the BSC is mainly high energy electrons, which signals, bulk signals depend on atomic number. X-rays is a longer recording time are needed. So when a scanning electron microscope is used, the electron optical column and the sample chamber must always be at a vacuum. If the column is in a gas-filled environment, the electrons will be scattered by a gas molecules, which would lead to reduction of the beam intensity and the stability. So it's very important that why you wanted to use vacuum. Because the electrons, if there is, air, the travel of electrons, generated from tungstens, before it go and hit the samples, it will be, affected the travel path. Also, the other gas molecules which could come from the sample or the microscope itself could form compounds and condense on the sample. So this would lower the contrast and obscure the detail in the image. So another one is important is the chemical and the thermal stability, which is necessary for well functioning the filament. Also, the field emission, the gun requires 10 to the power of 10 tares. So the signal electrons must travel from the samples to detector chamber pressure. That is also important. So the vacuum also requirement is depend on the type of detector, whether backscattered or secondary electrons. So it is important that, here we are going to deal with so many, samples, and we are going to see higher magnification, like a 300,000 times magnifications. So at the same time, when you are increasing the magnification, the resolution should be maintained. So the best resolution that can be obtained, size of the electron spot on the sample surface. So the volume from which the signal electrons are formed defines the resolution. Also, it's very, very important the scanning speed. So the electrons start from the tungsten filament and it's a travel to go and hit the samples. So in between the column, there are condenser lenses, objective lens that will control the path of the electrons. So once it go and hits, it come back and detect from the different detectors, and you can see the images, compositional and other images. So now, how quick it will go and hit and come back. So that is the most important thing. A weak signal, that means if that electron is a slow travel, that is not good. Also, too fast also not good. The optimal speed is always good to improve the signal to noise ratio. When doing a slow scan drift in the electron beam can affect the accuracy of the analysis. So today we can stop now here. Then tomorrow we will discuss next week with the, how we can process the samples for scanning electron microscope. Okay, see you next class.